pull this up. All right, let's finish up with the eye. And then we'll do the ear, the vestibular system, and then we be done. Okay, so if you recall from last week, we were talking about the different layers to the eye, the different tunics. And so let's start off with the outermost layer, the fibrous tunic. There are only two components, the cornea and the sclera. I'm going to show each of those individually. The arrows are pointing at both. All right, so the sclera is the white portion of your eye. And your conjunctiva covers the sclera, but it does not cover the cornea. Something to think about. And there's the cornea. Now the cornea does offer a little bit of refraction of the light. It will do a little bit of bending of the light. So as soon as the light starts to travel in, all right, the cornea is going to bend a little bit. You'll get a little bit of a slight bending in the aqueous humor. The lens does considerable amount depending on what it is that you're looking at. And then, of course, the vitreous humor will contribute a little bit too. All right, our second layer, all right, to the eye, our second tunic, all right, the vascular tunic. That's going to include the iris, pupil, ciliary body, suspensory ligaments, lens, and the choroid. The choroid is this brownish part all back here on the posterior portion of the eye. All right, so let's look at the iris. That's the colored portion of the eye. Thank you, melanin. That's what gives us the color. All right, as it does to the color of our skin and to our hair. Melanin is great and it's wonderful because it protects our DNA. Here's the iris on this other model right here for you. All right, so that's the iris, and then obviously in the center of the iris is the pupil. Okay, that is where the light is going to enter into, then pass through the lens and go back to the retina. So there's the pupil. And there are two muscles in the iris that are going to contribute to the shape of the pupil. And I love them because they tell you exactly what they do. All right, there's the dilator pupillae and there's the sphincter pupillae muscle. Real easy, easy stuff. You don't have this in your book, and I apologize, all right, but you do. You should have this on your Quizlets here. All right, this is showing you a picture of the ciliary body. Okay, ciliary body is this kind of pinkish structure here. Then you'll see the suspensory ligaments. All right, the suspensory ligaments attach onto the ciliary muscle there. And so when the muscle contracts, those ligaments become slackened. When the muscle relaxes, those ligaments become taut or tight, and they manipulate the shape of this structure, the lens. Okay. The lens is transparent. It is going to refract and focus that light that enters into the eye, and that will shine onto the retina. Here's the choroid, back portion here. You'll notice there's a lot of blood vessels in and around that structure there. And you'll also have some lymphatic vessels there. Remember, this is called the vascular tunic for a reason, and that's because there are blood vessels, lymphatic vessels in that layer. Speaking of layers, let's go to the innermost layer. All right, the retina, also known as the neural tunic. So that includes the retina, the macular luta, that's the structure that surrounds the fovea centralis, and then the optic disc, which is nicknamed the blind spot because there are no photoreceptors in the optic disc. So here you can see the retina, this pinkish-like structure. This is where we are going to find your photoreceptors. Here you can see it on this model here. All right, so there's the retina. This picture you don't have in your book, but this, I just want to show this to you. This is an ophthalmoscope image. So when the doctor looks into your eye and shines that light into your eye with that thing that they put up against their eye, where you guys get nose to nose. All right, this is the picture that they're looking at. All right, they're looking at several of those structures there that I will show you now. All right, they're going to be looking here at the macula luta. and the foveo centralis. So the macula luta is going to surround the foveo centralis. The foveo centralis is that little kind of yellow button in the center. Here's the macula luta. It goes around like that. All right, so that's the macula luta. Here it is imaged on this picture here with the red circle. And then at the center of the macula luta is the foveo centralis. That's where that's where your cones are going to be located. You do have some rods in there, right? But that's the sharpest image. Remember, cones are for a sharp, crisp image. There's the blind spot, the optic disc. That's where the blood vessels enter into and exit from the eye. Also, that's where the optic nerve is formed. All the axons of the ganglion cells are going to converge there and form the optic nerve. Right, and so that structure is the optic disc. Here you can see it as a whitish structure on the model here that should be in your atlas. That's the optic disc. Here it is on this model here, our optic discs. Okay, and there's a picture of the optic nerve. You can see it exiting posteriorly from the eye, cranial nerve number 2. Number 2 because you have 2 eyes. That's the optic nerve. All right, what the arrow is pointing to is this space here. That's the posterior cavity. Our reference point is the lens, which is not here, but this is where it would be. Everything behind the lens in that cavity is the posterior cavity. It is filled with the vitreous humor. It's the permanent vitreous humor because it develops when you are developing inside of your mommy, and it stays there. It doesn't circulate like the aqueous humor does. There's a picture of the vitreous humor that should be in your atlas there. Right? The vitreous humor is kind of like a gelatin, not thick like jello, but it helps to give the eye its shape, helps to keep the retina pushed up against the other tunics there so it doesn't peel off. What the arrow is pointing to here now is our anterior cavity. So again, here is our lens. So it's going to be everything in front of the lens. Right? And so the anterior cavity is filled with the aqueous humor. This is a circulating material, and so it's going to be produced and then it's going to be drained off. It gets produced, right, in the posterior chamber. So you can see here, the anterior cavity is pretty much from this line all the way to the cornea. Right, that's the anterior cavity. So our anterior cavity is divided into 2 chambers. Right. At that point, we use the iris as our reference point. So everything from the iris up to the cornea is going to be in the anterior chamber. Then everything from the lens up to the iris is going to be the posterior chamber. The posterior chamber is where we produce the aqueous humor. Right. The ciliary processes do that. And then it travels through the pupil. All right. Into the anterior chamber here, and it drains out, all right, through our scleral venous sinus in either spot there. And that is it for the eye, everybody. Let's quickly go ahead and do the ear. And then a tear will drip down my cheek, produced by my lacrimal gland, stimulated by cranial nerve 7, because lab will be over. External ear, all right, is going to be made up of 3 components: the pinna auricle, all right, which is that outer portion. Then you have the acoustic auditory canal, or the external acoustic meatus, and then the tympanic membrane. So there's the pinna or auricle. That's what collects the sound. Then the sound will travel down the external acoustic canal or the external auditory meatus. And it travels down that structure, and then it'll come in contact with your tympanic membrane. All of that makes up your external ear. Okay, so the pinna, the external auditory canal, and then the tympanic membrane. Then you have the middle ear, which is made up of the eustachian tube, the little bones there, the auditory ossicles, and then the tensor tympani muscle, which will be located in here in this middle ear space. Let's take a look at each of these. There's the eustachian tube, also known as the pharyngeotympanic duct. This is going to connect your pharynx, specifically the nasopharynx, to the middle ear, right? This tube here helps to equalize pressure across the tympanic membrane there. Also, it's a great place for a middle ear infection. Unfortunately, for some folks, if they get a throat infection, sometimes those little germies can find their way into the eustachian tube and work their way up into the middle ear there. All right, let's take a look at our auditory ossicles. The first one is the Malleus, also known as the hammer. All right, the Malleus is the auditory ossicle that attaches directly onto the tympanic membrane. The Malleus articulates with the incus, also known as the anvil. And then finally, the third bone, the stapes, articulates with the incus, and the stapes is also known as the stirrup. That definitely looks like a stirrup if you've watched Westerns or ridden horses. And so it's the stapes, the foot plate of the stapes, which is this structure here, that sits right against the oval window. And when these fellas vibrate, they will create those pressure waves in the scala vestibuli there. All right, the tensor tympani muscle, this is the muscle that helps you to pop your eardrum when you're trying to equalize that pressure. All right, it also helps to stabilize when I say stabilize, it will restrict the movement or excessive movements of the auditory ossicles when a loud noise occurs. That is the pediis muscle. Which brings us on to the inner ear, folks. The inner ear, also known as the labyrinth. We have the cochlea, that's the hearing apparatus. Then we have the vestibule and the semicircular canals. That's going to be your equilibrium or vestibular apparatus. So let's look at both here. Again, you can see we've got our cochlea, the vestibule, and the semicircular canals. Let's start off with the cochlea. That's a pretty easy structure to identify. That's the one that looks like a snail shell. Remember, it's just one long tube that we just rolled up in a horrible fashion. Okay. So you can see it now from the other side, and you'll see all these sensory neurons traveling. We'll talk about what branch of the cranial nerves that's going to be. All right, on the inside of our cochlea, sits on the basilar membrane, right, which is the floor of the cochlear duct. We have the spiral organ. In the spiral organ, we have our hair cells. And you can see, all right, the hair cells attached onto, actually, this should be moved. Whoops. Hold on. Un momento. These are your hair cells. All right. Wrong spot. No, much better. Right. So you have the hair cells here. These are the outer hair cells. These are the inner hair cells. Here are the stereocilia. All right, and they're embedding into the tectorial membrane here. All right, so let's look now in the cochlea. We're going to unravel our cochlea, and then we're going to take a transverse section right through it. And you'll see there's 3 chambers in there, and there's a couple structures in addition. All right, so the roof to the cochlear duct, this is the vestibular membrane right here. That is the roof to the cochlear duct. In the cochlear duct, we have our spiral organ. This is the sensory structure that allows us to hear. It's also known as the organ of Corti. All right, then going back here to the cochlea, all right, the vestibular duct is that upper chamber, also known as the scala vestibule. Right, that's where those pressure waves will come in from the oval window, and they'll travel down through this duct. Right, that's the vestibular duct, also known as the scala vestibule. Down below, all right, is the tympanic duct, known as the scala tympani. So once those pressure waves move through, all right, our cochlear, they go from the scala vestibule down here into the tympanic duct, through this area, and then they'll travel through the tympanic duct of the scala tympani towards the round window. All right, there's the tectorial membrane. The stereocilia of the hair cells are going to be located and embedded into the tectorial membrane. All right, there's the cochlear duct filled with that endolymph, that nice potassium-rich fluid. So when those tip links open up and open up the potassium channels, the potassium from the endolymph can diffuse right into our hair cells and cause depolarization. All right, the floor of our cochlear duct is the basilar membrane. Our spiral organ sits on top of that. The basilar membrane. Okay, that's it for the cochlea. Now let's get into the vestibular apparatus here. You've got the utricle and the saccule. Both of those structures are called the vestibule. The vestibule is going to be responsible for static equilibrium, all right, and then linear acceleration. So let's talk a little bit about the vestibule. That top sac-like structure is the utricle, and then the sac that it sits on top of is the saccule. So the utricle is on top, the saccule is down below. And both of those are called the vestibule. Here's a picture of the oval window real quick. Right, you'll notice that that's a perfect shape for the foot plate of the stapes bone to articulate with. So that's the oval window, and then down below it is the round window. Pretty simple when you look at it, right? One's round, one's oval. So you'll notice here on the left part of our screen, we have these semicircular canals, these loop structures. So you have three of them, right? This top one here is the superior or anterior semicircular canal. Right, and then we'll go down to the bottom one, and that's the posterior semicircular canal. Then the one that's partially obstructed there and kind of comes off at a weird angle, that's the lateral or horizontal semicircular canal. All right. At the base of each of these semicircular canals is that little bulge there. That's the ampulla of the semicircular canals. Remember the crista ampullaris and the cupula, those are located in there, right? And that tells our head that angular rotation, we're turning our head from side to side. Right? That information is collected there. So when we're looking here at the exterior of the structure that we're looking at, this is called the Bony Labyrinth. So that looks ivory to me, or kind of whitish. So when the arrows are pointing to the white part, right, excuse me, that is the Bony Labyrinth. When it's pointing to the gray part, that is the Membranous Labyrinth. All right, the membranous labyrinth is the gray portion, the white is the bony. Easy to remember because bone is white. All right. So again, when we're talking about our vestibular apparatus, everything that has to do with equilibrium, it's inside of that circle. That includes the semicircular canals and the vestibule. We call that the vestibular complex. And then there's the vestibule one more time. That includes the utricle and the saccule. And that neurological structure heading towards the cochlea, That is the cochlear branch of the vestibular cochlear nerve. Now you can also write in C, capital N, Roman numeral 8, which is a V with 3 capital I's afterwards, like this. Okay, you don't have to write it out. If you want to, you can, or you can do it like that, cranial nerve 8. So that is the cochlear branch of cranial nerve 8. And then we have the vestibular branch of cranial nerve 8, or the vestibular cochlear. Well, what? That's it, folks. That concludes